Good evening. In this lecture, we're going to talk about mosquito surveillance and determining the population of mosquito in a certain geographical area. So we're going to start out just watching a quick video on the Collier County surveillance program.
Hi, my name is Adrian Salinas and I work for the Collier Mosquito Control District in beautiful Naples, Florida. These aircraft are ready for another mosquito treatment mission. However, before they fly, we have to collect important data. Let's see how that works.
The Collier Mosquito Control District Surveillance Program collects data on mosquito species and relative numbers using the methods you're about to see. Inspectors hunt for mosquito larvae in fresh and saltwater habitats. The age and numbers of the larvae are noted. A landing rate count determines how many mosquitoes can be attracted in 2 minutes. There are about 60 landing rate count locations. New Jersey light traps attract and capture adult mosquitoes to monitor for species and relative numbers. There are 60 New Jersey light traps permanently stationed. CDC traps also collect adult mosquitoes and have the advantage of being mobile. All the surveillance data is collected daily to help determine when and where to treat for mosquitoes.
So that's how it works. The Collier Mosquito Control District and you, partners in public health.
So that is just how mosquito surveillance is carried out in Collier County. Broward County, Miami-Dade County, Palm Beach County, all the Monroe County, all of these different areas all have very similar methods of mosquito surveillance. So the whole point of surveillance is to monitor the mosquito population in a certain geographic location. We need to define the nature and extent of any kind of mosquito problem that may be occurring. And since mosquitoes are the most prolific of the vectors, disease vectors carrying parasites, bacteria, and viral diseases, it's really important that we monitor the mosquito populations in areas where mosquitoes are prominent. The other thing that mosquito surveillance is used to determine is whether or not a control program is actually working or not. If we see this sudden, really rapid increase in a mosquito population, then that's telling us that the efficiency, or what's called the efficacy, of a current mosquito control program isn't necessarily effective. So we're trying to determine if the program's effective. If it is, do we still need that program? Maybe we stopped that program for several months and we see that there's no longer an increase. Maybe it was water area needed to be cleaned up, or cleaning up a certain alleyway, or a clean your yard campaign, things like that that are part of control programs. Maybe that program was finally permanently successful and we can end any kind of chemical control method because we've gotten rid of breeding areas. The data from all of that surveillance, again, is combined to give us the kind of information that we will need to determine whether or not control methods are actually being effective. So the first thing we have to do in determining a surveillance method is to define if there's even a mosquito problem to begin with. Are we seeing diseases that are known to be mosquito vector diseases? Do we have so many mosquitoes that it's such a horrible nuisance that people maybe don't want to visit an area? Particularly here in Florida, we go through really extensive mosquito control mechanisms and control programs because we're a tourist industry. And if the mosquitoes are so bad down here that tourists and people from out of the state don't want to come and visit, we lose a lot of money. So economically, that could be a major impact. So it's important for all of these counties to invest pretty heavily in mosquito surveillance programs. So there's a major economic factor. The other thing we have to look at in surveillance is we have to define the parameters. What is this control program based on? Is it chemical control? Is it cleanup control? Is it habitat control? What are we going to be using to try and control this mosquito population? And we have to figure out what are the most appropriate survey methods for that geographical area to identify when and where to actually implement a control program. Just the mere presence of mosquitoes does not necessarily mean that there should be a control program. But if we have an excess of mosquitoes and we're counting those mosquitoes in the correct areas to get a true picture of what the mosquito population is like in those areas, then we can then determine a proper control program if needed. So in Florida, there are about 75 different species of mosquitoes here in Florida. 25 of those 75 species require trapping at night. So light traps are a really important part of any kind of surveillance or control program. There are about 24 different species that are considered problem species that could possibly be disease transmitters or vectors. Any of the 75 species, however, are considered a nuisance. So just because a mosquito is not necessarily a vector for disease does not necessarily mean that it's not a problem. The nuisance problem is a really big factor in areas like South Florida here, where we depend economically on a lot of tourism. So economic issues, so loss of tourism, property value. If you're living in a really swampy area where the mosquito population is really super high, that nuisance factor is going to become an economic factor in the value of your property. There can be loss of property development. There are areas where maybe a developer gets to an area and says, it doesn't matter what mosquito program I put in here, these mosquitoes are too awful. People aren't going to want to live here. We can see a reduction in livestock production, such as loss of milk production in dairy cattle, weight loss in beef cattle and in poultry, nuisance in poultry, and bloodletting causing the loss and death of very small birds in particular. The other is, of course, the spreading of disease. There was concern a couple of years ago when the Zika virus was in Miami. Miami was considered kind of ground zero for the Zika virus here in South Florida. So the spreading of diseases is, of course, of the most concern, but is not necessarily the major factor. Oh, sorry about that. So when and where is the actual problem? We start identifying a mosquito problem in a couple of different ways. The first and most common way is by complaints. So there are mosquito control centers in Miami and, of course, in Broward, Dade, those areas. And so people will call and begin complaining, and they'll say, hey, the mosquitoes are really awful this summer. Can somebody come out and spray? And if the mosquito control center gets enough complaints in a single geographic area, they'll come out, they'll do a surveillance to see what's going on with the mosquito population in that area. And it isn't until they actually do a little bit of their own data research and figure out if there is an actual population problem or if it's just somebody has a pool in your neighborhood that hasn't been cleaned or something like that. Those areas, if they're going to need surveillance, they'll complete some surveillance properties. Some people can complain online, so they'll use what's called the online mosquito spray request. And what's really happening is people in neighborhoods are having a really tough time, a nuisance factor with these mosquitoes. They're going to start complaining to the Mosquito Surveillance Center. The surveillance center comes out and they'll spray. But these areas they really do not like to spray in populated areas very often. So it really takes a big problem for them to come out and actually start spraying. Now, there are two different types of surveillance that are completed in addition to the people monitoring, right? So we have the people factor, them calling in complaints, but we have adult surveillance and larval monitoring. In adult surveillance, we look at three different things: what's called a resting population. This would be where mosquitoes are looked for during the day, and they look for these mosquitoes in dark areas, and they call it a resting population. Then we have the landing rate count, which you saw in the video. Literally, a person stands in a single area for 2 minutes and counts the number of mosquitoes that land on them. And then you saw a few examples in that video of some mechanical traps that are also used to attract adult mosquitoes. In larval monitoring, they'll go to the breeding sites, which are usually water sources. Mosquitoes lay their eggs near water, and the larvae develop in water. Larvae you can identify a species based on the different types of larvae that may be present. So they'll go to a breeding site inventory where there may be water sources. They will also do what's called emerging surveillance. So they'll have traps for the larvae, and they'll see how many larvae are emerging on a regular or daily basis. Right here is the information for the Broward County online mosquito, the Broward County Mosquito Control. You can use that number if you have a mosquito complaint. So in resting adult populations, adult mosquitoes are pretty inactive during the day, particularly in the heat of the day, mid-afternoon, noon, that sort of thing. So they will actually create what are called nesting stations. They are these large boxes, and mosquitoes like to nest or sleep inside very dark, cool, kind of damp places. They like to kind of go in there during the day when it's really hot out, and they'll just kind of rest and sleep during the day, and then they're much more active at night. So they'll create these nesting stations, and the nesting stations are used to perform counts. So they create these big boxes like this. You can see here in this section right here, it's all laid out, and inside of the box is lined with a black felt like fabric and sticky tape. And so the box is lined with all this sticky tape, and when the mosquitoes come in and they land on the sticky tape to rest, then they get stuck to the sticky tape. And then after a few hours, they'll pull the box, open it up, and start counting the mosquitoes that are on the sticky tape. So this is referred to as a nesting station. Natural resting stations are things like this bucket. What they do is they use these special types of vacuums, and they'll go inside of buildings and corners and they will vacuum out these areas looking for those adult mosquitoes. And they have a specialized filter on the end of this right here that will trap those adult mosquitoes, and they can then count the mosquitoes that they vacuumed out of a certain area. So natural resting stations are actual areas that mosquitoes would normally find. Artificial resting stations are these types in A and B up here, these types of traps that are laid out to mimic a dark, cool area for the mosquitoes to rest in. Now, animal baits in landing or biting counts. Sometimes animals are actually used for bait, and the most common are chickens. Chickens are very often used for baits. Sheep or goats can also be used, and they put them inside of this large tent here. So inside this tent, there's a funnel here where these mosquitoes can get into the tent, but then when they try to get out, they get trapped. And so they'll get trapped in these big vents here. And this is referred to as a Magoon trap. And in the Magoon trap down here, inside of the Magoon trap will be an animal, whether it's a chicken or a goat. Some type of small animal will be placed inside that trap to try and recruit or bring in the mosquitoes. We want to attract the mosquitoes, We want to attract the mosquitoes to that animal so we can trap them inside the Magoon trap, and then the next day they would count. Then there are entrance-exit traps, like the lard can. And so in the lard can, this works with this funnel type of structure here as well. There's a small basket that fits down that will have bait in it. So there'll be like a mosquito bait, chemical bait of some kind inside of this little trap here. And mosquitoes can go in through these funnels this way to get to the bait, but then when they try to get out, they get trapped inside the lard can. So it was called a lard can. You can look at the lard. Let's see if we can get this link to work and see the video on that. Nope, our link is not working, so we'll come back to that. Next is a landing rate count, and in a landing rate count, we're going to count the number of mosquitoes that land on a person in a 2-minute time frame. The mosquitoes, most often in the video, you saw the guy was just kind of brushing the mosquitoes off and I guess keeping a count on his own. But more often than not, we not only want to know how many mosquitoes are landing on the person, we also need to know what species of mosquitoes they are. So in this case, the aspiration method is used most often. It is most effective, the landing rate count is most effective for salt marsh mosquitoes and what are called container mosquitoes. Those that like to breed in old tires or buckets, anywhere there's standing water in some type of container. Aspiration is done with these small little jars. This is called the collection tube, and this one is a manual, right? So this is a manual aspirate. And you would actually suction, there's usually a mouthpiece on the end here, and there's a filter. So you would actually suction with your mouth or blow, and that would push the mosquito through the filter and into the collection tube. Over here is one that's used more often nowadays. This is a really pretty cheap one. You can make your own aspirate tube if you need to. Over here you have an aspirate gun. It does the same thing. So you can walk around and just pull the trigger, and it will suction those mosquitoes up into a container right here. And the mosquitoes will get trapped inside of the container, and then you can take it out. And both of these can be used in the landing rate. So we look at how many mosquitoes, and they'll aspirate those mosquitoes to get a count of the landing rate and of what different species of mosquitoes are actually in that landing count. In mechanical traps, we have a couple of different types. We have what's called the New Jersey Light Trap. The Jersey Light Trap is a permanent fixture. You can see it uses quite a large bulb, and it has a big funnel, and then it has these catch containers here at the bottom. Inside of here is a fan, and so the mosquitoes are attracted to the light, and the fan suctions the mosquitoes down through this funnel into this collection tube here. This is how most light traps will work. So here they're showing you, here's the fan. It's a very gentle type of fan. It's not a strong suction or anything, but we have this light bulb. The mosquitoes come in, the airflow is this way. So the mosquitoes get pulled down through the airflow past there, the fan here, and down into this collection vessel here. There's little funnels, a screen funnel in that, and that's to funnel the mosquitoes down into that collection tube. Sometimes they'll put a paper bag with cyanide granules in the bottom of it, and the cyanide will kill the mosquitoes so that the mosquitoes don't try to fly back up out of the trap. And then the bottom of the container after 24 hours will have the mosquitoes in it. Jersey light traps are used for permanent surveillance. So if there's specific areas that a surveillance center wants to watch on a regular basis, they want to check this once a week, or during mosquito season it might even be every day. Those areas they're going to install a Jersey light trap. So it's on a wooden post. They're very large and very heavy, and they are used most often for permanent surveillance in specific areas. The CDC light traps come in a couple of different flavors. We have a white light trap and we have a UV light trap. UV light usually attracts some different mosquito species than the white light. We have both of these in our labs here at Broward. We use both the white light and the UV light trap. There's usually a battery pack that's located somewhere in here. It's in this little motor here. This one would have a battery pack probably up top in here. Ours is up top. Takes 1 or 2 D-cell batteries. They're good for about 12 hours, 10 to 12 hours. Now that battery pack is going to run both the light and the fan that's found here. But instead of having that big heavy metal area and this really big giant trap like we saw with the Jersey trap, The CDC light traps are very light. They're very easy to move around, so they're portable, which makes them really great for looking at different areas. They come with these little containers here at the bottom that has a screen, so there's an oxygen, there's airflow, and the mosquitoes get attracted to the light. The fan blade, that's the fan right there. The fan will be turning. This will draw the mosquitoes down into this net bag and into this container. They don't usually put cyanide granules in the CDC light traps. That's used for the Jersey light traps. You can see in this one here, it's the same thing. The mosquitoes are getting attracted down into that. We're just using UV light instead of white light. Now we look at oviposition traps. And oviposition traps are used specifically to look for Aedes aegypti and the Culex mosquitoes. Here they will take water with oviposition cues. So they'll have maybe some stagnant water. They'll take water from a local pond, water that would attract mosquitoes, and they put it here in the bottom of this kind of big bucket type of thing. And then on top of the water, they put netting. And this netting just sits on top of the water, and this is to help promote oviposition. The water usually are some lures in here, and there's a clear container that sits on top of the water, and the clear container has a funnel-like hole in the top of it, and then a funnel is placed into that clear container, and it funnels eventually really much more narrow than that down into the water. So what happens is these female mosquitoes are attracted to the stagnant water, to whatever cues are put in there. They come down to the mesh, and the mesh is sitting on top of the water. They can land on the mesh and lay their eggs along the mesh. Then when the female tries to leave, many of them will get trapped in this clear container here as opposed to moving back out. In doing this, we can sample the eggs, and we can also let this sit for 24 hours and then take it back to the lab, allow those larvae to begin developing, and we can take a look at, get an idea of how many eggs are being laid by how many females. Gives us a general idea of the population in oviposition. Now in true larval monitoring, this is carried out mostly in urban areas because we want to make sure with standing water, if there are mosquito larvae in that standing water, we need to, of course, get rid of it. We also want to use the larval monitoring for early indicators of a change in species in a geographic area. GPS mapping is most often used to map out different breeding sites. So these could be lakes, ponds, swamps, pools. You might have an entire neighborhood and all of a sudden there's these mosquito problems. You go through the neighborhood, you see that there's a bunch of abandoned houses, and maybe some of those houses have pools. If those pools are left unattended, they become gigantic mosquito breeding grounds. That was a really big problem during here in South Florida during the housing crash several years ago. Many of the houses had pools, and if those pools are not being maintained, the chlorine will eventually dissipate out of the pool, and we're left behind with just plain water. That's an awful lot of water in a big in-ground pool. That water will become stagnant. Algae will start growing, it gets stagnant, you get frogs start getting in there, and of course you get mosquito larvae. So sampling immature larvae, we use a dipper, and this is a big white cup with 2 grooves in it, and it's attached to a big long extending pole. The pole can extend anywhere from 6 to about 10 feet, depending on the type of dipper that you have. Most of them are about 6 to 8. The dipper, you skim and you partially immerse the dipper in the water, and that will look for the Aedes mosquito, or you will completely submerge and that looks for the Culex mosquito. So you can also, in smaller areas, if you don't want to use these big dipping cups, in smaller areas you can use a pipette or an eyedropper. When you collect the larvae, you would take a look in here. You get a general idea of the age depending on the size of the larvae, and if they're floating on the top, we know that they're an Aedes. If they're submerged, we know they're Culex. So we can start using a GPS system to start mapping where we're seeing these larvae in big numbers. So that gives us an idea of where these different breeding areas are. Other methods for larvae, dipping doesn't work for all species. For the Aedes aegypti mosquito, it works for some of them, but not all of the different Aedes species. Wyomia and Culiceta. You can use a meat baster, you can use a bilge pump, or you can pull up aquatic plants. Some will actually lay their eggs along the stems of aquatic plants underwater. That's what's going on here. These are larvae that are attached to plant material underwater. So you would need to pull up the plant material and then look for the larvae actually attached to plant stalks in water flora. Now, CDC recommends surveillance to monitor levels of these different factors. They want to look at virus activity. The University of Florida monitors virus activity in mosquitoes here in Florida. So they have mosquito traps set up all over the state. They oftentimes will get samples from the different mosquito districts in the different counties, and they all are sent to a central lab in Central Florida where they test them using PCR for different viruses. They also look at the number of species of mosquitoes in different areas because they are looking for different vectors of different diseases. The Aedes aegypti carries yellow fever, but a different mosquito might carry Zika. So we want to know what species of mosquitoes are out there, in what population numbers, and where. Are there any infections in humans? We want to know if there are any current humans infected with diseases that are vector-borne from mosquitoes. Do we have other vertebrates that are infected with vector-borne diseases, such as birds, dogs, cats, any other kind of warm-blooded mammal? So in CDC recommended surveillance, we look for any kind of vector-borne disease in all vertebrates, but we look at those in humans separately. The other thing that is kept track of is weather factors. Is there anything going on weather-wise that might change the growth rate of a certain type of mosquito, or change in a weather pattern in an area of the state or in the country that would cause a change in species? Maybe one area is receiving more rain than normal, and so mosquitoes that wouldn't normally grow in that area or increase in population in that area because of the arid quality of the area, if it's getting a lot of rain and a lot of bad weather, we could see a rapid increase in different species of mosquitoes. Surveillance is usually used to make sure we want to detect if there's low immunity in hosts. We want to know if there's an excess or what we call a vector abundance. And we're also looking to see if there's any kind of favorable condition that would promote epizootic or epidemic type diseases. So the Zika outbreak here in South Florida was considered a small epidemic. Epidemic and outbreak are oftentimes used interchangeably. Now, that was an epidemic. We had an increase in a population of mosquitoes in that area. We had a disease that was imported here into the state and then later found in the mosquito population. So the disease in the human is one thing, but when the disease is not usually found in an area, and then all of a sudden, not only do we have a human with that disease, but we now monitor our population and we see the activity of that particular virus in the mosquito population, that changes everything, because that now means that mosquitoes are accessing that particular disease. It does not mean that mosquitoes are going to spread it to each other, but it means that we could possibly start seeing community transmission of a vector-borne disease in a small area. And that was the big concern with the Zika virus for women who were pregnant. So there are 64, let's talk a little bit about a few of the diseases that we surveil. So there are 64 different arboviruses found in vertebrate hosts, and all of them have some kind of effect on human health. And all of these viruses that are listed here are all transmitted by mosquitoes. So we have Eastern equine encephalitis, St. Louis encephalitis, Western equine encephalitis, La Crosse virus, West Nile virus, Zika, chikungunya, malaria, yellow fever, and dengue. Yellow fever is now in Florida. Malaria and dengue are the only two diseases that have not been found in Florida in the mosquito population. We have had cases of dengue here in Florida, but they are what are known as imported. So somebody picked up dengue somewhere else, and then by the time they got to Florida, they became symptomatic. So it's not considered a domestic case. In sentinel animal surveillance, that was that animal that was put in the Magoon trap that I talked about earlier. These are called centene or sentinel animals. So it's sometimes referred to as sentinel surveillance. In Florida, we use a domesticated chicken. Blood is taken from the wings every 2 to 4 weeks and tested for the presence of arboviruses as well as tested for the presence of antibodies. So when that chicken is put out into that Magoon trap for a few hours, once it's taken out of the Magoon trap, it's then quarantined for about 2 weeks. And after 2 weeks, then blood will be taken from the chicken and they'll look for certain different viruses, the presence of viruses. They will also look for the presence of antibodies. So this is a way to determine if disease is actually being transmitted by these mosquitoes. They can trap free-ranging birds. We catch them in nets. The birds would be bled usually through the wing, and then they'll take a blood sample. That's all that means. Then the bird's foot would be banded and the bird is released back out into the wild. Horses are also very often surveilled here in South Florida, and of course that's because we have 3 different forms of equine encephalitis here in South Florida. Rabbits, squirrels, chipmunks for lacrosse virus, and raccoons also for a multitude of viruses. But the horses are primary hosts in those equine encephalitis diseases. So horses are very often surveilled for mosquito diseases. Now, if human arboviruses are reported, it's reported to the DVBID. I got to remember what that means. That's the Division of Vector-Borne Infectious Diseases at the Centers for Disease Control. That's what DV, this is, Division of Vector-Borne Infectious Diseases. There are a bunch of specialized forms that have to be completed. Of course, this needs to be diagnostically sound decision. So the diagnostics are based on the signs and symptoms of different diseases. Of course, any kind of serum testing or CSF testing. CSF is cerebral spinal fluid. That's because many of these viruses are neurological viruses. And then are we looking at an acute stage of the infection? Is the person currently exhibiting signs and symptoms and they're very sick? Or are they in the convalescent stage of infection? Convalescence is really the recovery stage, is this after infection. Now this makes a difference because in convalescence we're going to look for antibodies. And in the acute stage we're going to look for a viral load. So the different laboratory methods that are used to surveil these different diseases, we can use what's known as direct or indirect antibody fluorescence. So if antibodies are present, we can attach little fluorescent molecules to those antibodies and look at them under the microscope using a fluorescent microscope, and that's going to tell us if we see the glowing, then we know that those antibodies are present. We can look at something else called hemagglutination inhibition. This is a coagulase test. And so we're looking for coagulation in a blood sample based on the presence or absence of different antibodies. Another is called a fixation test. This is also known as agglutination. It's very similar to blood typing. And if antibodies are present or the virus is present, depending on how we're looking at it, if agglutination occurs, then our test result would be positive. If no agglutination occurs, then it's considered negative. Neutralization test is carried out in a test tube, and we can neutralize the ability of a virus to enter into a cell based on antibody production. ELISA tests are used for either viral load or antibody load. So we can use ELISA tests. Those are the most common. That's what they're using right here. This is a form of ELISA. So viral load or antibody load. And then the most common nowadays is PCR. Polymerase chain reaction is looking for virus genes. It's a way to genetically search an organism to look for a very specific gene sequence or genetic sequence that is unique to that organism. PCR and ELISA tests are the most common. ELISA, these are your rapid tests. So in a rapid test like we have up here, this is a form of ELISA. A pregnancy test is a form of ELISA. So the liquid sample is put into a well over here, and it starts to run up this filter paper, and the filter paper has antibodies embedded in it that are specific for the pathogen you're looking for. So if that pathogen is present in this liquid sample here, as it moves up the filter paper, it will come into contact with those antibodies, and it causes a color change to occur on that filter paper. PCR is done with a computer and a thermal cycler. It's a much more extensive test, but it can be completed in anywhere from 2 to 3 hours, and results are really very accurate. ELISA results are great looking for whether you're looking for an antibody or an antigen. They work really great. They're very quick, 5 to 15 minutes max, but they are sometimes considered not as reliable. So more often than not, those rapid ELISA tests are going to be considered preliminary, and then they'll confirm the presence or absence of a viral load using PCR. Now, in natural disasters, when natural disasters occur, FEMA starts to ask the CDC to start looking if and determining if our arboviruses are actually going to start spreading. And arbovirus, by the way, means arthropod-borne virus. That's where that name comes from. So arbovirus is not a family of viruses. It's just any virus that is arthropod transmitted, vector transmitted. After natural disasters, because of the loss of infrastructure, of course, wreckage and damage to buildings, those sorts of things, places are usually a mess after some kind of natural disaster. Mosquito populations do have a tendency to increase. So the CDC considers the following different characteristics of a natural disaster, or I should say of surveillance of these arboviruses for determining the risk of the transmission of disease. So has there been a change in the mosquito population index? Are we seeing increases in populations in certain areas, in all areas? Is it focused in one specific area? What are the infection rates of the viral infection rates within the mosquitoes themselves? So we need to set up traps and then bring those mosquitoes back and run PCR on them to find if they're carrying different viruses. Determine if there's increased transmission in vertebrate amplifying hosts. So let's take a horse, for example. A horse is a really big animal. So if an equine encephalitis virus gets inside this horse, it's going to replicate exponentially, and it's going to be producing millions and millions of copies of it, far more copies of that virus than, say, a mouse. Evidence of disease in horses is usually a really big indicator. It's difficult for mosquitoes to bite horses, so when we start seeing disease in horses in particular, that means that we're seeing vector transmission. They'll take into consideration, of course, rainfall and temperature information. Is it hot and muggy, or is it cool and dry? What's the time of year, and what is the risk to humans? Are we locating this in a horse farm that's 100 miles from anywhere else, or are we seeing this in a horse farm that's in the middle of downtown Davie, where we have thousands of people living close by? So the CDC has a set of definitions of risk for arbovirus diseases. The first, we're going to go in order of severity. So the first is referred to as the negligible outbreak of disease. This is usually when there's some kind of vector found, but it's off season, and so it's inactive. The climate is not favorable. There are no corrections need to be done. There's nothing that we're going to do. We're just going to leave that area alone. If there's a remote risk of outbreak of disease, it might be spring, summer, or fall. And within those 3 different seasons, we do see mosquitoes of varying populations. The adult vectors are very active, so the adult mosquitoes are active, but in low populations. And of course, larval development requires warmer temperatures, so the temperatures may still be cool enough that larval development does not really occur very quickly or doesn't occur at all. And oftentimes what they'll recommend is some kind of source reduction. So in source reduction, they'll put insecticides in water or larvicides in the water, and of course continue the surveillance. Here in South Florida, we are usually under a constant remote outbreak of disease. So the CDC is basically saying, you've got enough mosquitoes and there's a little bit of virus here and there in the population. So it's not a bad idea to make sure that you put insecticides in those public water sources, and that you clean up areas and take just preventative steps. Possible outbreak of disease means that we have a kind of a localized, referred to as focal abundance of a particular vector. We start seeing an increase in a very specific species of mosquito, and that mosquito is a known vector of disease. In addition to that, we know the temperature is now good for larval development, so the mosquito can increase in population very easily and quickly. We start seeing what's called seroconversion in our sentinel hosts. In seroconversion, what's happening is instead of looking for viral load, we now look for antibodies. Antibodies are produced in response to a viral infection. So if we have a sentinel animal that now has a high antibody count, this means that at some point prior they were infected with that particular virus. In this case, in possible outbreak, the CDC recommends increasing local treatments, start using what are referred to as adulticides. These are a little harsher type of insecticide, and increase the surveillance. So that usually constant surveillance would be a Jersey light trap. When they start to increase their surveillance methods, they'll start putting up those temporary or portable CDC light traps. Then we have probable and outbreak in progress. A probable outbreak means that we have a whole population of adult vectors. We have a confirmed case in humans or horses. We have a great survival environment for larvae. Larvae are able to survive very well, and they're in their best conditions for development. Also, the vector survives. And if this is the case, then the CDC recommends these 5 things here: implementing emergency control, putting adulticides in areas that are high risk, putting larvicides in areas that are known breeding sites. This is when you start hearing from the CDC to warn the public. That's what was going on in the very early portion of the Zika outbreak. And then surveilling hospitals to see if any human cases of that particular disease show up. Finally, the outbreak in process this occurs when we see multiple confirmed human cases, which happened here in South Florida with that small Zika outbreak. We continue emergency measures with the adulticides and larvicides, but now we start to hold daily briefings on a regular basis, kind of like they're doing with the COVID virus right now. This is something that would have been done. This was done with the Zika virus. Every day there was some kind of news conference telling the public what was going on with that particular disease. And of course, they will continue surveillance for human cases in local hospitals. So that's all we have in surveillance of mosquitoes. It's a pretty straightforward lecture, and I will get the